or no? I've taught you. Okay. So in that chapter, you people have seen how different groups of microorganisms are so detrimental to the health. They cause so many different kinds of diseases. But now, in today's video, you're going to see how the same exact group, how they can be useful to us as well. So that's the case. So microorganisms, they are one of the most major components of our ecosystem. They are very tiny, not visible to the naked eye and everything, but it's that they play a great big role as being a part of the ecosystem. So microorganisms, what are the categories? We have bacteria, we have fungi, we have virus, protozoa, and these are abundantly present everywhere, right? The thing with these microorganisms is that you can find them everywhere. They're found in the soil, they're found under the rocks, in the rocks, they're found in the hot springs, like they're found in those areas where life is not even possible at all. So you find them in extremes of the conditions. Now, so basically, these microorganisms, they can be grown by us. You know that you, me, anyone, if we get hold of bacteria, we can grow the bacteria. How? You people have seen Petri dishes in your school, your lab, Petri dishes. Everyone has seen? That's the rest of the students. Switch on your videos, please. So when we talk about the petri dishes, so how do we grow microorganisms? For example, I'm taking the case of bacteria. So in a petri plate, you add all sorts of nutrients. That is called as the media. Okay, the media is the nutrient that the particular microorganism will be utilizing to eat and to grow. Then you take a spoonful or a loopful of microorganism and sort of just spread it over the media. You keep it for 24 hours and then you will observe that on the piece of the petri dish or on the petri dish side, you'll see several circular, fluffy or white or blue or red structures that we call as a colony, okay, colony of microbes. What are the media that could be used? There are some examples like nutrient agar media, potato dextrose media. These are the media that are used. Now further, when we talk about the microorganisms, they are useful to us in variety of the fields. What are those fields? First is the household products. Microorganisms are very much utilized in the household products and their production. Second, industrially, they are also important. Sewage treatment and also in the production of biogas. So when we talk about the microorganisms, so they are used as biocontrol agent, they are used as biofertilizers also. Everyone knows about fertilizers. In general, what fertilizers are? Is there anyone in the class who doesn't know what fertilizer is?
Enrich the soil fertility.
Yeah, they do that. So everyone knows what a fertilizer is. Now you people also know this fact that those chemicals that increase the soil fertility, apart from increasing the soil fertility, they also cause soil pollution, isn't it? They cause water pollution as well. So they're not very, you can say, useful in the long run. But biofertilizer, use of living organism as a fertilizer. So will it cause any harm? No, it will not cause any harm, but it will cause only positive effects. So first, we're going to see how the microorganisms are used to make household products. Now this, the first one to everyone has had. Is there anyone who's lactose intolerant in my class? No one. Okay, good. So none of you are lactose intolerant. But even so, if anyone is lactose intolerant, they can have curd as well. Now, curd, everyone has had curd? You know what curd is? Okay, good. Everyone, you people are not aliens. Nice to know that. Okay, so the curd that we talk about, it's nothing but a product from a bacteria. Now imagine when you're eating curd, you're eating millions of bacteria. So how is curd made? You people have seen your mothers making curd at home? How does she do it? Can anyone tell me?
Mom, we had vinegar.
Vinegar? No,
Yeah,
that
we
is
make cheese. That's cheese. Sorry, that's cheese.
that is cottage cheese. Yeah, that is for paneer. No, not that. How is curd made?
With
You
milk
make
and
a fresh milk with some other curd or something.
You take milk and then you add spoonful of curd that you already have at home, right? Then you cover it and you keep it overnight, and then you see in the morning you have freshly made curd. Now these days there are refrigerators that have that system in them that can make curd for you, that provides the temperature and everything. Now, when we talk about curd, so it is formed from the partial digestion or coagulation of milk. And that is done by the lactobacillus bacteria, which is lactic acid bacteria. So during the curd formation, what we do? We add a culture or a spoonful of bacteria, spoonful of curd. That spoonful of curd has enough bacteria that you put into the milk, and then that bacteria will start utilizing the milk as a substrate, and it will digest the milk partially, which becomes the curd. So the bacteria that is there, which is lactobacillus. And if you provide them, and you people might have seen that it's not very easy to make curd during winters. No one has it also. Like no one consumes curd during winters. But apart from that, it's very difficult to synthesize curd during the winter. Why? Because the temperature is low. And during that temperature, lactobacillus bacteria would not be able to work. So it requires a suitable, optimum temperature, suitable temperature, and then it will multiply and convert the milk into curd. So that is how it is made. And the process of formation of curd is called as curdling. Right? This is called as curdling. Now the question comes along, which one do you think is better? Is curd better or is milk better? And whoever says whichever answer, I need to know the reason why. And not the one that I've written. Which is better, curd or milk?
Curd.
Acha, why is curd better? Why is it better? That's true, it's better. But why? Okay, now you people will understand. Curd is more nutritious and it is much better than milk as it contains more vitamin B12. While the milk is being converted into the curd, at that point of time, the vitamin content of the milk increases, particularly vitamin B12, which stands for cyanocobalamin. Lactic acid bacteria. This LAB stands for lactic acid bacteria. So these are also found in the stomach to keep our stomach disease free. So the first product that we get from microorganism, which is a part of the household product, is the curd. Is it clear to everyone? Any doubts are there?
No, ma'am.
After that, we have the next, that is cheese. Acha, how many of you like cheese? How many of you are here like the cheese lovers? Let's see. No cheese lovers? None of you like cheese? Acha, Jesslyn does. Acha, first of all, to everyone switch on your face. I'd like to see your faces. So keep your videos on. Okay, so you people don't like cheese. Why, students? What has cheese done to you? Why? Has it done something bad? Or did you have too much of it that you're now so over it? Oh, ma'am, nothing, ma'am. We can't answer. It's a very private matter. Eh? It's a very private matter. You people can't tell me. It's okay, Jesslyn. I understand you love it. Keep your hand down. Okay. Now, moving forward, talking about the cheese, that is also a product of microorganisms, different microorganisms. So again, cheese is also a partially degraded concentrate of milk and milk protein. Now, how many cheese varieties you people know? Let's see. How many cheese varieties? I just speak now. When I'm talking also, you people don't want to speak. How many cheese varieties you people know? Or you people have had? Let's see. Acha, everyone might have had mozzarella, cheese pull. Everyone has done that. Okay. Then what about
Cheddar cheese.
cheddar cheese is also there. Then
A
anything
cream
else?
cheese.
Which one, Jobita?
Sliced cheese, the burger one.
Sliced cheese is the only. It's like a way it is presented, but that also comes in a lot of different cheese variants. Like there's ricotta cheese, and then there's cottage cheese. Everyone has had paneer, right? Everyone has had paneer, so that is cottage cheese. Then what else? Acha, how many of you have had those? Is it called? Mozzarella is done. Cheddar is done. Ricotta
Feta
is done.
cheese for
Feta cheese,
salads.
yeah. Yes, very good. Feta cheese is there. So yeah, there's so many cheese varieties. Now imagine for different cheese varieties, there are different microorganisms playing a role. So the cheese, like, and you might have had, like, if you've had all the varieties of cheese, you people know how they vary in texture. And some of them are very stretchy. Some of them are extremely salty. Some of them are very slimy, right? So it can vary, and the fragrance also varies. Apart from that, the flavor also varies. So this varies because of the microorganisms. So the few examples that are there in your book. So there is Swiss cheese. Have you seen that classic cheese picture? That's like the Tom and Jerry cheese. You seen that? The ones that have big holes in it and it's like a triangle. So that is your Swiss cheese, which is made by the bacteria called as Propionibacterium shermani. So this is the bacterium which is responsible for the formation of Swiss cheese. Then there's another kind of cheese called as Roquefort cheese. This is a cheese that is made by the fungus called as Penicillium roqueforti. And lastly, there's the camembert or camembert cheese, which is made by Penicillium camemberti, which is again a fungus. So bacteria and fungus both are involved in ripening of the milk to form the cheese. So that's the second product we get from microorganisms. After that, there's bread, dosa, and idli. Acha, how many of you have had dosa and idli? Had, so everyone has had. Who loves dosa and idli? Even I do. Okay, Jaslyn does. What about, okay, Safrina does. Then, who else? Muniba also does. What about Aisha? You don't? Marwa, you don't? No.
Mom, in my place, we don't eat this.
Acha, you don't eat this. Acha. Okay, what about Jobita? Acha, Jobita also does. Okay, so majority of the members do. Acha, Malwa, what about? Acha, Malwa also does. Okay, nice. Okay, okay. Now you people can put your hands down. Okay, very good. All right. Even I love it. So now these, acha, you people might have seen if these things are prepared at home, your mother basically, keeps it like the rice batter or the batter of, this thing, semolina or any kind of batter that is prepared. It is usually kept overnight, isn't it? And once you open it in the morning to see, you'll see large holes present in it, right? Or even for bread, people might have seen how in cooking videos also how they make the dough and then they cover it and leave it aside. And then they watch it back and then the dough has fluffed up. So how that is happening? So the fluffing up or the fermentation process is happening because of yeast, which is a unicellular fungus. So baker's yeast called as Saccharomyces cerevisiae, which is unicellular fungi. So basically, the baker's yeast is added and it makes the bread porous. Porous means a lot of holes are present and it makes the bread soft. How does it do so? Because during its, you can say, growth or survival, the yeast does fermentation. And during that time, CO2 gas is released, which causes the rising up of the dough. Then, same goes for dosa and idli batter also. Then comes toddy. Now, toddy is an alcoholic drink which is, used in the southern parts of India And it is made by fermenting the sap of palm tree. It's like a delicacy of that area. It's like a very famous thing in the southern parts of India. So that is again made by fermentation process. So you people understand this thing that microorganisms are capable of fermentation, which yields us a lot of products that way. So this is the case. Up until now, is everything clear?
Yes, ma'am.
Then comes, now this was the first set, that is usage of microorganisms in the household products. After that, we come on to the usage of microorganisms in industrial products. Industrial products means those products that are created in industries and they are sold on large scale basis, right, as a production in industry. So microbes, they are used to create beverages, meaning several drinks, and even antibiotics, the medicines on large scale industrially level. So basically, when you think about it, fermented beverages. Now, the best example of fermented beverages is the production of alcoholic drinks, like things you might have heard or seen in televisions. Also, there's wine, there's beer, right? There's rum. These are alcoholic drinks which are made by fermentation process. And during the fermentation process, the bacteria or the yeast, which is a fungus, is involved. Now, what it does is that it basically ferments the juice and creates alcohol out. Okay, so alcohol, which type of alcohol? So that was ethanol, which is ethyl alcohol. Okay, so this thing, see, fermented beverages. So yeast species used in alcoholic fermentation are Saccharomyces cerevisiae, which is also called as baker's yeast, as well as it is called as brewer's yeast. Brewer is the one who makes drinks or alcoholic drinks. So there are different types of yeast species that are used for different kind of drinks that are produced. The main thing is that the final production is of the ethanol, which is ethyl alcohol. And this is what is the content of the alcoholic drink. But one thing you people should understand, that yeast poisons themselves, is poison themselves as the alcoholic content, as the alcoholic concentration. Okay, alcoholic concentration reaches 13%. So this tells us that in naturally fermented alcoholic drinks, the concentration of alcohol will be only up until 13% and not beyond that. But you people know there are few drinks that people can have, like they take in tiny glasses, and once they drink it, they immediately become drunk. It means that particular drink has a higher alcoholic concentration in it. So how does it add when it's not possible to have 13% and more? So there's another process which those alcoholic drinks undergoes, and that is distillation. So all those beverages that are alcoholic, but fermentation will be done in them, which will not be followed by distillation. So their alcoholic concentration will be very less. Whereas the drinks that have fermentation followed by distillation, so those drinks are going to have a higher alcoholic concentration. For example, wine and beer, these two drinks are produced without distillation. So they have low alcohol concentration. On the other hand, whiskey, brandy, and rum, these are produced by distillation. So they have a very high alcoholic concentration. So if you see in this particular table, so there they have shown you how the different alcohol types are there and the concentration of alcohol they have. So beer has the least, that is 3 to 6%. Wine, 9 to 12. Whiskey has 50% alcoholic concentration. Brandy has 60 to 70%. Rum has 40%. Gin also has 40%. And lastly, there's vodka, which has the most amount of alcoholic concentration, that is 60 to 80. This much is clear to everyone, students? Any doubts? No doubts. See, this is the picture of a fermentation plant. So the things like any raw material, like the fruit juices, molasses, the wheat and barley juices, all of those things are put into it. And then anaerobic condition is created. Anaerobic means the one that does not have enough oxygen. So the first industrial product are like the fermented beverages. Is it clear, students? After that, we have the antibiotics. Now, antibiotics are those chemicals that are made in order to help us to survive any kind of disorder or to kill the microorganisms that are present in our body. So, those antibiotics are the chemical substances, right? And, antibiotics, they are a chance discovery. How? See, while Alexander Fleming, he was working with a bacteria called as Staphylococci. So see, whenever you work with bacteria, you have to be very careful so as to not contaminate. Now, he saw that when he used an unwashed Petri plate, on that mold grew. Mold, you understand? Fungus. Fungus grew on them. So he saw that on the plates where the fungus has grown, this Staphylococci bacteria was not able to grow because of some many reason. Like nutrients and everything was provided, but the bacteria was not at all able to grow. So he found that the chemical that was produced by the fungus was penicillin, and the name of the fungus was Penicillium notatum. So this was the discovery. It was a chance discovery that Alexander Fleming did. And its full potential as effective antibiotic was established by Ernst Chain and Howard Florey. So these three people, they were given, like they were awarded with the Nobel Prize in the year 1945 for this very discovery. And this particular, like the discovery of antibiotics have helped to to combat or to treat so many deadly disorders or diseases. And during the World War II, I believe, or World War One, it was used, uh, World War II. So it was used to treat the American soldiers that were wounded during the World War II. So imagine antibiotics are also produced by microorganisms. Now they produce it as their defense so that nothing can harm them. But us humans, we're so intelligent, we utilize that for our own benefit. And it works well for us. So antibiotics are clear, students? So that is. Do you people have any idea about any other chance discovery of anything? What all things are discovered by chance and they were not supposed to be discovered initially? Any idea? No idea. I'll give you one thing. Do people eat cornflakes? Cornflakes milk? Those cornflakes, they were not supposed to be cornflakes. It was a chance discovery. It was something that was not actually supposed to be a thing. It was nothing earlier. What happens is that the dough with which the cornflakes are made, like the maize dough and stuff. So when whoever was the worker, he took the dough and he microwaved or he put it in the oven for a long period of time. He by chance forgotten it. As a result of which the whole entire dough or the particles they became really hard. Right. So when once he picked it up, he sort of applied a little pressure, it broke down into several pieces. He was like, okay, this is bad. Now we'd have to throw it. So then he picked it up and then ate it. And then he thought, why not if we can eat this with milk? So that is how cornflakes came into existence. It was nothing. It was not even such an idea. It was a chance discovery. Same way, penicillin is also a chance discovery, for which these three people were awarded with the Nobel Prize. Now they their names are known everywhere. Whenever we'll study about antibiotics, we'll know that. Now, antibiotics have greatly improved our capacity to treat the deadly diseases such as plague, whooping cough, diphtheria, leprosy, that earlier used to kill many, many people because there was no cure. There was nothing to help them. But then, because of the development of antibiotics, these things have been greatly used. Any doubts up until now, students?
No, ma'am.
Okay. Now, furthermore, from the microorganisms, there are a lot of enzymes, chemicals, and bioactive molecules that are also synthesized. So, bioactive molecules are those molecules which are functional in living system and they can interact with their components. So, microorganisms are again used to make them, such as organic acid, alcohol, enzymes, cyclosporin A, and statins. This table is very, very important. This is something you people must learn by heart. That is, the microorganisms used, the product they produce, and the use of those products. The first is Aspergillus niger, which is a fungus. So it produces citric acid. Now, citric acid is used in dyeing, inks, it is used in medicines, flavoring, and also used in the preservation of food. Second is Acetobacter acetate, which is a bacterium and it is used to make acetic acid. What is acetic acid? It is basically a fancy name of vinegar. So it's used for the production of vinegar. Then Clostridium butyric. It is responsible for the production of butyric acid, which is used for making rancid butter. Lactobacillus makes lactic acid used to make curd. And lastly, the penicillium. So that is gluconic acid, which acts like a great calcium source to lactating mothers or infants and even cows. Clear? So these are the various microorganisms, the products they produce, and the use of those products. Then talking about apart from the bioactive molecules, we have the enzymes that are produced by the microorganisms. So the total enzymes like lipase. Lipase are the enzymes that are usually, like they are used in detergent making so that they can remove the oily stains from our clothes. So the microorganism from where the lipases are obtained are Candida lipolytica, Geotrichum candidum. Then pectinases and proteases. Acha, students, have you noticed the difference between the juice that you get in a Tetra pack from the market and the juice that you've squeezed at home? Can anyone tell me what's the major difference among these two juice? Like, for example, you've taken out orange juice at home, and orange juice is like one, it is store bought. Can you tell me what would be the major difference?
More
Tell me.
sugar.
More sugar, correct. Then
Preservatives.
preservatives, yes, preservatives are added. Also, you might have seen how the juice that you take out at home is very, you can say it has pulps also. It is thicker. But the juice that you get from the market, it is very smooth and very clear. So, pectinases and proteases, they help in clarifying the fruit juices to make them clear. And the ones that we have at home, they're not that clear, right? Microbes used for the production of pectinases are Aspergillus niger, then Bisoclummis fulvo. Proteases are obtained from Mortriella renispora, and Aspergillus and Bacillus species. Now, proteases are those enzymes that degrade proteins. Then, some other products that are made are streptokinase. Now, streptokinase is produced by the bacterium called as streptococcus. And you people know that, like people that have undergone heart attack, right? Or that are at the risk of heart attack, they are because their blood vessels have some sort of clot, isn't it? So this enzyme, that is streptokinase, which is made by Streptococcus bacterium, it is very much helpful in removing the clot from the blood vessels, like arteries and veins. The last enzyme is amylase. So what they do is that they degrade the starch and they are obtained from Aspergillus, Rhizopus, and Bacillus species. Any doubts up until now, students? Anything you people want to ask me? After the chapters like evolution on molecular basis, this might be sounding like a story, isn't it? This might not be as boring, I believe.
Nice.
Then we have cyclosporin A. It is another chemical which is prepared by microorganisms. So this is immunosuppressive agent. You people remember how those people that have undergone organ transplant, they have to stay on immunosuppressants all their life because their immune system will attack the organ. Graft rejection, CMI. Yes. So these cyclosporin, these are the chemicals. So these are the medicines that they have to take in order to suppress their immune system. And this cyclosporin A is made by Trichoderma polysporum. The last one is the statin. Now these are produced by the yeast called as Monascus purpureus. Now they are blood cholesterol lowering agent. They lower the cholesterol present in the blood. Okay. And they basically, how is the mechanism? It acts by competitively inhibiting enzyme responsible for synthesis of cholesterol. So it doesn't let that enzyme only being synthesized. Clear, students? This part you don't have to learn. It's not important. This much is clear up until now. The use of microorganism in industry and in household, both of these things are clear. Okay, very good. Now you people note down, then we'll move forward and talk about how the microorganisms are used for sewage treatment. Okay, the next topic.
Amina.
Done, students? Shall I scroll down?
Yes, ma'am.
Done, students? Shall I scroll down? Shall I scroll down, students? Yes or no?
Yes.
And students, shall I scroll down?
Yes, ma'am.
Everyone's done? Then students, shall I scroll down?
Yes, ma'am.
All right. Everyone's done? Do you people have any doubts up until now?
No, ma'am.
Done, students?
Yes.
Yes, ma'am.
Okay, write the cheese part and then we'll start the next topic, that is sewage treatment. Done, students?
Yes, ma'am.
All right. Okay, now let us see the next topic in which the microorganisms are used, and that is for the treatment of the sewage. Now, what is sewage? It is the municipal wastewater, meaning anything, the wastewater from the homes, from the hospitals, from the industries, that is constituted under sewage. So, large quantities of wastewater is generated in cities and towns. The major component is human excreta, then rubbish, the waste, and everything. Apart from that, it contains a large amount of organic matter and microorganisms as well, a lot of which are pathogenic. They are obviously not useful. So now usually these, like the sewage that is generated, now they, like the sewage is directly discharged into the water body, which obviously pollutes the water body. So thereby, to prevent the pollution, what must be done? The sewage that is created or the sewage that is collected, that sewage must be, you can say, treated before it is dumped into the water body, so that there is no pollution that is caused. So the treatment of the sewage water is done by the heterotrophic microbes that is present in the sewage only. This treatment is carried out in 3 steps: primary treatment, secondary treatment, and tertiary treatment. So when we talk about the primary treatment, so this is a physical process, okay, wherein all the floating matter, all the soil particles, all the floating debris is removed. So large particle removal is done during the primary treatment. So first, sequential filtration happens, in which the floating parts are removed, and during the sedimentation, the larger, like the soils, the stone, the pebbles, they are allowed to settle down at the bottom of the tank, that is called as sedimentation. So this is the primary treatment. Now all the solid that settle down in the like they become the primary sludge. And the supernatant means the floating part, that the part that was at the top becomes the primary effluent. Now this is the wastewater, right? It still has microorganism, it still has waste particle. So the primary effluent is now sent for the secondary treatment. Now, in the secondary treatment, what happens is that the primary effluents, they are passed on into large tanks that are aerated, aeration tanks, meaning constantly air is pumped into this tank. Now, because the air is being pumped, so aerobic bacteria starts growing in this tank and they become flocks. What are flocks? Flocks are net-like structures or mesh-like structures of bacteria and fungus. So while growing, these microorganisms, they start consuming the organic matter which is found in the wastewater, and they start utilizing that organic water. As a result, the wastewater does not contain organic matter, or the organic matter starts getting reduced at that point of time. So when this is being done, so it reduces the BOD, which stands for biochemical oxygen demand of the effluent. If the BOD is reduced, it means that the pollution is also reduced. So what is BOD? It is the amount of oxygen that would be consumed if all the organic matter in 1 liter of water were oxidized. So it is a measure, the amount of oxygen that can be taken up by microorganisms. So if the BOD increases, it means more pollution. If the BOD is less, it means less pollution. Now the sewage water is treated till the BOD is reduced. And after the BOD is reduced, then the effluent, the part from the aeration tank, is sent to the settling tank, where all those flocks that were formed, they are allowed to settle down or allowed to sediment. Now, this sedimented part is called as activated sludge. Now, this activated sludge is sent back to the aeration tank to act like the inoculum so that the process keeps on going. The rest of the part is sent to the anaerobic sludge digester. Now, in the anaerobic sludge digester, the methanogenic bacteria, meaning those bacteria that have this capability to produce methane. So, those bacteria now start digesting the sludge and produce methane gas, hydrogen sulfide, and carbon dioxide, which is called as the biogas. Now, this biogas is inflammable, meaning if you light it, it can catch fire, and that is why it can be used as a source of energy. Is it clear, students, up until now?
Yes, ma'am.
So now further, the effluent of the secondary treatment, meaning whatever was left, now it can be released into the water body because it does not have any polluting substance in it. But for example, if you want to